Just as with prokaryotic cells, a eukaryotic cell is filled with a semi-fluid jelly-like substance called the cytosol, and scattered throughout the cytosol are found many different structures called organelles. Organelles, again, are unique to eukaryotic cells. We've gone through a list of those structures, but here we'll go through them individually, discussing them more in detail. We'll begin in the middle of the cell at the nucleus, where the cell houses its DNA. The membrane of the cell is referred to as the nuclear envelope. It encloses the DNA, physically separating it from the rest of the cell. The envelope itself is actually a double membrane. The two membranes, each a lipid bilayer. These two bilayers are separated by a small intermembranous space. The envelope is also regularly perforated by these pore complexes, called nuclear pores. And at the lip of each of these pores, the inner and outer membranes of the envelope are continuous with each other. The DNA found within the nucleus is primarily in one of two forms. One form is the tightly bound chromosome, which is probably the most familiar form and is typically found in the nucleus of an actively dividing cell. The other form is more of a diffuse mass, referred to as chromatin. This is how DNA typically appears the majority of the time in a non-dividing cell. Also prominent in a non-dividing nucleus is the nucleolus, which is a denser mass of DNA where ribosomal RNA is made and the proteins for ribosomes are assembled into large and small subunits. These ribosomes are then exported through the nuclear pores out into the cytosol. The nuclear pores also serve to transport mRNA to the cytosol, where it's translated into proteins by those ribosomes. Many of the proteins made within cells are secreted into the extracellular space, but the process of making that finished protein began at the endoplasmic reticulum.